Okay, so our guest speaker for today is Mike Brudno. I'm sure many of you know. So Mike is the Canada Research Chair in Computational Biology and Associate Professor in Department of Computer Science and here at the Rna-Seq. He is also the Director of the Center for Computational Medicine at Sick Kids Hospital. And his study background is having done his BA in Computer Science and History. I'd like to figure out how you put those two together. At UC Berkeley, he went and did his PhD in computer science at Stanford, working on algorithms for whole genome alignment. Michael completed his postdoctoral fellowship at UC Berkeley and was a visiting scientist at MIT before coming to Toronto. And Mike Brudno, as I'm sure many of you know, his main research interest is in the development of computational methods for the analysis of genome data sets. And I'm sure many of you who were in the room 3 till 4 heard that he is the recipient of the Ontario Early Researcher Award, the Sloan Fellowship, and Outstanding Young Canadian Computer Scientist Award. So many accolades. Okay, great.
Thank you. Just a laser point. Good. So I guess one of the things I have to say is thank you for having me. It's actually, I've already given a talk at MonBug, and I sort of kind of gave a talk at VanBug. It wasn't really a VanBug talk, but it was correlated, so it's good to go for the trifecta. In George Costanza's words. It's really a pleasure to speak at now TorBUG. Also, the talk you're going to see is actually not my sort of standard canned talk, because I was going to put some stuff together and just take my old talk, roll it out one more time, but it turned out that I got scooped by my own graduate student who actually presented a good portion of it here a couple of months ago. So you're going to get something completely different, and we'll all see how it goes. I will start with sort of a few reasonably canned slides just to set the setting. And all of you have probably heard about the high throughput sequencing data and the fact that the data is exploding all the time. And if you've survived all these explosions, you know that we started out with really big reads. Well, really big. Now they look really big in retrospect, but you could get very few of them, and over time we got more reads but fewer, and now even more reads but they're even smaller, and we're trying to figure out how to make sense of these next generation sequencing or high throughput sequencing technologies, these short snippets of genomic or of genomic data that we can generate from a biological sample and then try to analyze and try to learn some biology about the organism from which it came or about the tissue from which specific genes were expressed. So, in case you're not really that familiar with high throughput sequencing, I have a one-slide summary of it. Basically, the key idea is that you're going to be doing lots of sequencing experiments in parallel. You have a slide, you attach DNA molecules, and you're going to sequence by hybridization. As new nucleotides attach, you're going to have some light emitting for whatever the tag that was attached at that position. And this basically starts looking like a picture, and you end up taking lots and lots of these extremely high resolution pictures over time as new nucleotides are added, so that you can actually reconstruct the actual sequence. So maybe in the first picture you got a green dot here and a blue dot there, a green dot here and a green dot here, so both had a T. Here there was maybe a blue and a green, so a G and a T, and so on. At each location, you over time record the signal. Because you're doing lots of these experiments in parallel, you can generate lots and lots of individual read sequences. The problem being is because there are so many, there are problems in terms of them being too close to each other and other things where you're having issues with the quality of the data, and also you can't really go that far. You can't make really long pieces. So what are this stuff useful for? Well, it actually turns out it's useful for quite a few things. first of all, obviously sequencing of genomes. So you can use these high-throughput sequencing technologies to take a genome of an individual and reconstruct it and try to understand what's causing a genetic disorder, for example, or how are humans in one population different than humans in a different population. It's used for expression data analysis. So by sequencing RNA rather than DNA, we can go back and try to reconstruct the actual isoforms of genes that were present in a particular sample, and also the expression of the genes, both at the same time. So this was really a revolution in terms of you did not have to know what you're looking for as you did with the old microarrays. ChIP-seq, where you cross-link the DNA with protein, and you do immunoprecipitation to identify where proteins bind onto DNA. And there, what you will be looking for is where are just lots of reads coming from, because they are probably proteins we're binding, has allowed us to learn a lot about which regions of DNA are regulatory. So there was a lot of news recently with the ENCODE project using these technologies extensively to learn a lot about human regulation of human genes. And pretty much every single time you turn around, there is some new kind of seq data, and the input is some kind of thing that looks like DNA, and the outcome is a very happy scientist who has published a paper in Nature or Cell or wherever they like to publish it. And so in order to do this sort of pipeline, there are several things that we'll need to be able to do. We need to be able to actually have the machines to sequence the genomes. We need to have bioinformatics analysis pipelines that can get you from the raw data, from terabytes of individual nucleotides and quality values, to something that can be interpreted by a geneticist or a biologist or a clinician. And finally, we need to have actually an interface for the clinician to look at the data and to understand it. You don't want to give a geneticist just a list of a big file which looks like this and then say, have fun. You want to be able to present the data in a compact and intuitive way. So this is actually, I think there's more and more there is a shift to an interpretation and analysis challenge. So we need to be able to do these steps much more efficiently. The sequencing data is becoming higher and higher quality. It's not, there are still issues, but it's becoming less and less problematic, more and more standard. The bioinformatics analysis and the visualization are what are the bottlenecks. So we need better pipelines to analyze the data and need better visualization methods to view the results. And today I'm going to talk about sort of two interrelated projects in my group. And if I have time, I'll get to a third one where we have tried to develop both interpretation tools and visualization tools and merge the two of them together. So by the way, I want to make it as interactive as possible. Please feel free to ask questions if you get as you have them. Don't wait until the last slide when you're completely and hopelessly confused. So the first thing I want to talk to you today is a tool that we've developed for Rna-Seq data analysis. And so this is, we're going to be looking at the mRNA, so the messenger RNA that's present in a tissue sample. And so similar to Leo, we want to sort of figure out how many of them there are in a particular sample, and also what are the actual messenger RNAs. Are there some new ones that we weren't really expecting? Unfortunately, we can't really read these directly. What we can do is fragment it, select to approximately a known size, and sequence read pairs. So sequences from the two ends of a fragment of RNA. And what I would like to do now is actually given this data, somehow figure out what the actual RNA was that was in my sample. And there are several things which are going to complicate the problem. The first and the most sort of obvious is alternative splicing. Well, just because you have a specific gene, you'll have various alternative versions of this gene. You could have an exon which is sometimes included and sometimes excluded, and it could actually be included and excluded within the same tissue set, within the same tissue that you're actually analyzing. 50% may have this cassette exon and 50% won't. Or 80% will not have the cassette exon and 20% will. And what we would like to know is where are these and what's the actual abundance of each one. There are things like mutually exclusive exons. There are interesting cases, things like intron retention, where a certain intron is retained in the mRNA and is being translated, probably leading to a horribly misformed protein, or just a protein which is going to be non-functional. There can be alternative. So this is actually intron retention is reasonable. It happens in several types of cancers and in other disorders. You can have alternative splice sites, alternative promoters, and other kinds of alternative splicing with various different polyase. And we would like to be able to reconstruct these. So the first step in order to do this is actually alignment. When we have the unknown mRNA and we generate read pairs, which are approximately a known distance apart, so the red, the yellow, and the blue pair, the first job is to take it and to map it. So this red pair would map nicely to exon 1 and would be an appropriate distance apart. The yellow pair will map to two different exons, and because of the intron that's in between, they'll now look like they're really far apart, right? And the blue pair will actually map like this. And this is called a split read, where a read goes onto two separate exons. And these are actually reasonably difficult to align. They're, you get lots of artifacts, and you can't always capture all of these, just because you're looking for a smaller segment to match up.
Do you actually map to the whole genome, or do you construct sort of a synthetic reference of all the mRNAs, of all the isoforms?
Yeah, that's a good question. Do we map to the whole genome, or do we map to construct synthetic mRNAs and map to all of these? So you can see that there'd be advantages and disadvantages to both. Mapping to the genome would have the advantage that you can capture novel things. Let's say there was an exon which nobody had previously annotated. You'd be able to align to it. But if you just look at the known mRNAs, you wouldn't be able to reconstruct. At the same time, mapping to the mRNA, this split read will be much easier to map, because now the two exons are going to be together. We actually do both. We
de novo assemble the transcripts, which I think is what you would.
That's de novo assembling transcripts we don't do. That's a separate problem. So yeah, but what we do is we map to the genome, get everything that looks like an exon, and try to predict what kind of transcripts there may be, then build libraries for the full possible set of transcripts and map to those. And that actually turns out to be one of the most time-consuming parts of the algorithm, mapping to all the possible transcripts, of which there are many more than all of the known transcripts. So Rna-Seq can give you better accuracy, base level resolution in SNPs compared to expression arrays. And the key thing is you're no longer restricted to what is annotated. So if there's a new exon which you didn't know about, new gene, you can actually measure it. If there's a new isoform, you can see it without having to design a probe specifically to capture it. But to really take advantage of it, we need methods that can use Rna-Seq data optimally. Here are some cases which we may want to capture. There's the novel, there's a cassette exon which is sometimes present, sometimes absent. Or, for example, there's an intron which is, in some cases, spliced in. And spliced in, it's not spliced out properly, so you're going to be generating a protein from it. And our goal is to determine all isoforms that are present in the sample and quantify each one. So there are tools that have been developed for one problem or the other. But the two actually are very, very related. You cannot quantify the isoforms without knowing which ones are there. You can't even quantify ones which you know are there without information about some which are missing. And vice versa. You cannot determine determine which isoforms are present without knowing their quantity. Some of the difficulties, Well, there's going to be lots of sequencing biases and variances that are going to make this problem much more difficult. Reads are not going to be sampled randomly from across the gene. If you look at the coverage signal, there's going to be biases in us being unable to capture reads at the borders of the genes, just because when you do the fragmentation, the pieces are going to be too small very often. We're going to have biases in that we can't capture the exons, the borders of exons as well, because of the sequencing issues. There could also be other biases, for example, a sequence-related bias. Maybe this area is particularly GC-rich, and as a result, the sequencing technology can't read it as well, so you will see less sequence.
Was the exon-exon junction bias one of mapping or one of actual sequence chemistry?
Mapping, yeah. It's a technical issue, but we still have to deal with it. There are other things. For example, there are PCR duplicates. So when you're doing the sequencing experiment, you do PCR, and you may end up over-replicating a specific fragment. So you over-sequence it, and as a result, you end up with lots and lots and lots of copies where there really should only be one pair representing all of them. So different tools have different, some people say, just throw all of them out. But then your quantification is going to be off because you're going to undersample in areas. Some of them say, you're going to the reason you're going to, if you throw them, you don't know what's a PCR duplicate. It could be a PCR duplicate, or it could be just you happen to capture the exact same fragment twice if your coverage is high enough. So you can't just throw them out, otherwise the really high coverage genes are going to be undersampled. And you can't keep them in because you're going to misestimate your coverage. They're going to be also, the other thing is pre-mRNA. This is something that most people ignore, but in reality, when you're sequencing an RNA data set, you're going to get some amount of unspliced pre-mRNA, which is going to actually end up being sequenced. So you will see coverage. The exact amount, sometimes it's as high as 30% of the actual expression of the gene will be the pre-mRNA. This things before splicing has ever happened. And we want to be able to subtract that out and to explain and to understand that this is happening. There are also difficulties with multi-mapped reads. So these are going to be reads which can come from many isoforms or many genes. So if this particular sequence, if this sequence is the same as this one, any read that you generate will have the exact same sequence. It could be from this isoform, could be from this isoform, or from that whole different gene, which just happens to have the same domain. So you are going to be interested in characterizing these. And most reads can originate actually from multiple isoforms. Some can come from different genes with shared domains. And figuring out which isoform a read most likely came from is a chicken and egg problem, because it depends on knowing the quantity of the isoform. So let's forget gene 1. If this isoform has twice the quantity of this isoform, this read is twice as likely to have come from here as from here. But at the same time, you don't know what's the abundance of each isoform until you figure out where this read came from, right? So you can see it's sort of a chicken and egg. You can't solve one problem without knowing the answer to the other, and vice versa. So all of these problems need to be solved by an algorithm to really to analyze RNA-seq data. So the algorithm that we developed and just recently published in Genome Research, it's called iReckon. I think everybody is getting tired of iProducts. It's going to be our first and last iProduct, I think. And it actually depends on considering the full space of possible isoforms and then quantifying all of them and getting rid of the ones which we don't think are expressed. So we start with this idea which is called the splicing graph. And the splicing graph tries to link together possibly adjacent exons. So here we have several things happening. We have split reads which join these exons, and also this to this one, and this one to this one. And here we have very high coverage within an intron, which sort of makes you think maybe the intron's being retained. So the set of isoforms is going to look something like this. This will be the splicing graph, and then there may be an intron here. The set of Parsimony isoforms is going to be, well, you could have this exon and no intron, have this exon and retain the intron, don't have the exon, and with the intron and without the intron, right? So 2 possible things, 4 possible isoforms. And you can see that the number of isoforms is actually going to grow very, very quickly with the number of possible alternative events. In practice, it actually turns out to be not too high, so we are able to actually enumerate all of these. And this goes back to Leo's original question. We initially map to the whole genome, but then we will map to the full set of possible isoforms for every single gene, for every single read.
But do you pre-annotate the exons? That's what
we pre-annotate, so we do 2 steps there. We take first known exons. We run a linear which can identify new possible exons which were not in our known data set. And all of these are going to go back into this model where we reconstruct the splice graph. We do, so the pre-computation, yes, you do it for every single data set. You have to. Yes, yes, because the set of possible isoforms depends on the actual data. How? No, to be honest, I don't remember. Bigger. At least the mapping time is much higher than the mapping time. The mapping time to the set of isoforms is much higher than the mapping time to the genome. Could be an issue because of the total number of isoforms, yes.
So if the median gene has, what, 10 exons, then it's to the 10,000 per...
No, so because we're not actually looking at so the question was, if the median gene has 10 exons, aren't you looking at 1,000 isoforms for the average gene? No, because the average gene doesn't have 10 alternative splicing events. If we don't see these links, if we don't see alternative splicing, we just join the adjacent ones. So it's going to be 2 to the order of alternative splicing events per gene.
So, yeah, but you still have a sequence for every isoform. Like you could have some string graph type approach where...
You could. So the idea that Leo is suggesting is a very good one. You could actually think of this as a string graph. And if you don't know what a string graph is, don't worry. And you could map to the string graph. Unfortunately, there aren't really out-of-the-box tools that will map to string graphs very well. So we have ended up, we do take some shortcuts. There are some places which we collapse together, but it still ends up being the most time-consuming part of our algorithm is actually mapping all of the reads to the full set of possible isoforms. So, yes. So to solve this chicken and egg quantification problem, we actually use something called the EM algorithm. So hopefully many of you are familiar with it. The idea is that we can estimate the abundance of isoforms using some assignment of the reads, but then use those new estimates to improve our abundance estimation and do this multiple times. There was actually several really nice papers which showed that under some completely unrealistic assumptions, this will converge to an absolute global optimal solution. In our case, we introduce other constraints which violate those assumptions. So in our case, we don't necessarily get to the absolute best solution, but we try multiple times from multiple different start sites in order to get to a good solution. So in the E step, we compute for every read the probabilities that it comes from a different isoform using the abundances computed at the previous iteration. And at the M step, we use these probabilities to recompute the isoform abundances. And I'll just do a little bit of a graphical visualization of this. Let's say that this is our read pair. This is the actual isoform, and this is the read pair that gene and the read pair that we generated. This read pair can map to any of these possible isoforms of the gene. But the likelihood is going to be slightly different. For example, if we map it to right here, the distance is probably going to be horribly off. So this is the pre-mRNA. Because it will include both, it will include all of the introns in between. So instead of being at the expected distance, the two reads will map much, much further apart. In other cases, let's say this is missing this intron, this other exon. So likely the reads will be too close together. So the likelihood will be a bit lower as well. So we can compute the actual likelihood for every single isoform that the read came from that isoform. So it gives us some kind of probability. And we also have some prior probability of which is actually equal to the abundance of that isoform. So this read is very unlikely to come from here, maybe very likely to come from here or from here, and a bit less likely from here or from here. And using these probabilities, we can assign the read to an isoform. So maybe we'll assign it to this one. In reality, we use soft assignment, but for soft assignments are hard to draw on a PowerPoint slide. Then we take a different pair, do a similar computation, have a different likelihood. So maybe these are the likely cases, blue, pink, or yellow, and equally likely. So we randomly assign it to one of these. Once we're done, we've taken all of the read pairs and assigned them to all of the possible isoforms. And we now have reasonably well estimated. So we have lots of reads which went maybe to these three isoforms, these two, fewer to these two, and very few to this one.
And the relative frequencies of the signs strictly based on distance?
They're based on distance, based on the quality of the mapping. So if you have more mismatches, it'll be. Yeah, I don't think we actually use base qualities, but they would be because they would go into the mapping quality. We use the read mapping quality, but the base qualities would enhance that. Yes.
Do you have like a data-based distribution of the distances? So like not every library is going to have the same sort of distribution.
We just look at basically what we do is we take all read pairs, map them to long exons, and learn what the expected distribution is. So given these, we can re-estimate the abundances of the various genes. So now this one we're thinking is actually has a higher abundancy, so does this one, and these two have lower abundances. And just to repeat, we now have this pair again, the very first one we started with. But now it's much more likely to be coming from the blue and from the blue because it's got blue has a higher prior. So we will just iterate this.
Will this find local maxima?
So right, so this is what I mentioned, that there was a paper that said that if all you care about are mapping qualities, it will actually find a global maxima because this energy space has a single maxima. And if you have only one maxima, it's still a global one. But in our case, it does find a local maxima, and we do multiple restarts from different configurations to try to minimize the probability of getting stuck in a bad local maxima. We can't guarantee that we give you the best answer, but hopefully it'll give you a good one. So this will be, the outcome. The next step that, yep.
Do you actually calculate the abundance? Do you take like average number of bases covering?
We look at the abundances are typically computed in this measure called RPKMs, which are reads per kilobase per million. So it's fragments or the number of fragments or reads which are mapped. We're using soft map.
Every mapped base is the same. It's more informative because you only have a single.
We're not looking at bases. We're looking at fragments which are mapped. The number of fragments. And if a fragment is mapped to two different things, we use soft assignment. So half to here, half to here. We don't look at per base coverage. Per base coverage gets tricky. And
you have more information if you have an exon only retaining one isoform, everything that maps there is sort of stronger than.
Yeah, I'm not sure if that's strong enough to actually take good advantage of, but yeah, it's a good point. So the second thing we do is regularization. One of the problems with EM is that it tends to find global is that it'll give everything in abundance. There's no reason to, if I had two identical isoforms, they will get each some abundance, and then they will just stay there. But they're identical. Why can't we just merge them? So there's this concept of parsimony, right? You want to use the smallest number of isoforms to actually explain your data. And this is done using regularization. Basically, what we will try to do is take reads which can be explained by highly expressed isoforms and take them from low expressed isoforms and move them to the highly expressed ones. But not all such reads, because if a read maps just to a low abundance isoform and not to any high abundance isoform, that actually tells us that probably it belongs to the low abundance one. So we'll be trying to do things like this. Move things which map well to high abundance isoforms. And then once we do a little bit more, we will end up with these quantities. This is, very low expressed, but these two are expressed, but these two are actually at 0, even though they had slightly higher abundances just before. So this is done using something called regularized DM. So we have basically we take the log likelihood and we regularize it with this extra term. If you're familiar with regularization, it's very common to use lasso, so linear regularizers, which actually turn out to work horribly here. The reason being is that these linear regularizer basically would try to subtract out the abundance of all the genes which are expressed. And the sum of the abundances is fixed. So if you think about it, we have this constraint where there's, the sum of theta I, where L is the length. So this is the likelihood, this is the length. The sum of all of them over all the isoforms is 1, because we're actually doing relative abundance estimation relative to other genes. And as a result, because of this constraints, basically doing lasso is doing nothing. You subtract out the constant. And what we end up doing is we subtract out a regularizer that's based on the square root, and the idea, well, actually the fourth root, but the square root is the idea being that if all things are equal, the sum of the square roots is going to be 2, while the sum of the square roots in this case is going to be 1. So this setting will be penalized more highly, and this will be the bottom one will be preferred. This is sort of a technical aspect. And we also have this intron retention model where we basically for every intron go through and try to compute what's the likelihood that it was actually retained, that it was present in our sample, even though at a much higher quantity than the pre-mRNA. So to do intron retention, you need to quantify the mRNA. And in this case, you will see something like this. So maybe in this intron is retained. So overall, the tool, if you compare it to other popular tools for doing abundance estimation or isoform discovery, there's lots of things which other tools don't. So it uses regularization, and unlike previous tools which use regularization, it does not do linear regularization, and it uses models duplicate reads, Pcr duplicates, models intron retention, and models pre-mRNA as biological features which you'd be interested in.
Can I just ask one thing?
Sure.
The assumption of at most one intron, what does it come from?
It comes from the idea that these intron retentions are going to be rare. It's actually not a good assumption. Just the space of isoforms explodes if you allow any intron to be retained. Then you get this aspect that if there are two exons, you get 2 to the 10th possible isoforms right away, because you could have any of the 10 introns or 9 introns retained. So it's a sort of a computational concern. How well does it work? Well, I wouldn't be here if it didn't work better. So these are the obligatory, our numbers are bigger than previous numbers slide. But if you look at, it's actually on simulated data, it has both higher precision and recall. Good sensitivity for novel isoforms, which are not given to the simulation, for exon skips, and for intron retention. previous tools, struggle with these. Also, it's if you look at abundance, at the accuracy by abundance, it does, compared to previous tools, much better at low abundance as opposed to high abundance. So high abundance means there's lots of this gene present. It's reasonably easy to figure out that it's there and to quantify it. At low abundance, it becomes much more difficult because you just have less data. And lots of previous tools, if they saw too little data, would just completely ignore the isoform. It's maybe noise. Because we use regularization, we actually let the likelihood determine everything for us. And we count on the regularizer to say all of these low abundance genes actually are present or aren't present. So it ends up doing very well. Here, the striped bars are things which are known, and the dark bars are things that are novel. So novel low abundance isoforms, you can see it's significantly better than previous stuff.
Novel compared to what?
So we give the program some number of annotated isoforms, not all, but some fraction. But in the simulated data set, there are both those and things which are not in the set of isoform given to the program. So those are novel.
So the novel between couplets and
They're the-
isolation might all be in the same set, or would they be distinct from one another?
They could be this. Well, it could be that some are a subset of these, but all programs are given a set of known isoforms. That's the same set, and all programs are given the same simulated set of reads, which come from either known ones or hidden ones, which the programs don't know about. So we want to know how good are the programs at determining the two types.
Do you wiggle the regularization parameter, or do you fix it up in the box?
We wiggle the regularization parameter. We sort of learn it over as we go through.
So how many known and unknown ones do you supply?
Yeah, I think we're doing on the order of 10% unknown, mostly picked from genes where there will be a different known isoform.
So basically, your data means that you can discover roughly 55% of the known ones, and you can discover half of the unknown ones.
No, it means that it's hard to compute from this plot what you want to do. Yeah, I know what you want to do, but it's trickier. I think it's for the unknown low abundance isoforms, we're in the 20% range. 20 to 30% are discovered, and that's much better than other stuff. Yeah.
Have you looked at if there's annotation errors on the training side? So if they're invalid exons?
So for our training stuff, we simulate everything, so there shouldn't be anything that's invalid, right? But yeah. And similar, if you look at abundance estimation, this is sort of real versus predicted versus simulated. I reckon does one of the best. Other tools sometimes are slightly off, and or have just a bit more dispersion around the XY axis. Let me just skip this. This is a real data experiment. What we have done here is we take a real RNA-seq data set, but when we ask the programs to compute on it, we hide some true human genes. So now we ask the program, how well do they find these new true human genes? It could be some of them aren't expressed. Many of them possibly aren't expressed. But on average, if you find more of the hidden genes, you're probably doing better as long as you don't predict too many other things. And it turns out that we get sort of better recall and much better precision using this. So we give the program a real RNA-seq data set, and we give it 80% or 90% of all human genes, and ask how well does the program do at figuring out the other 10%. So I want to do a really quick aside, and I only have 5 minutes, or even probably 3 minutes by now. I'll try to wrap it up really quickly. As part of doing all of this, we wanted to do both not just analysis, but also visualization of the data. So as part of the Savant Genome Browser, which many of you may have heard of, which is a genome browser developed in our group, we actually, which allows for plugins, we developed an RNA-seq data analysis plugin, which I will really, really quickly show you, because if I do it any slower, Michelle is going to chase me out of the room. So let me skip to right here. Within the RNA-seq plugin, can you see it on the screen? Yep. When you look at a specific region, it will show you the mapped reads. It will show you, including split ones. So in a second, it's going to go to a region of the genome and really quickly grab a gene that's in this region. It will show you the sets of exons. This is from UCSC. The abundance of each isoform. Each isoform is a colored line. And for every single read, it's going to show you the likely isoform from which it came. So these purple ones are actually most likely coming from the red or the blue, which are the most likely, the highest similarity ones. You can see that right here, there are blue and green, which start a little bit earlier than the red. So there's an alternative acceptor or donor site, depending on which way the gene's going, right there. So this, it's a plugin which you can run, I reckon, right within the Savant browser if you have it installed. You can just download it. So just to go back to my presentation. Yes. So just we looked at a few specific cases also. There is a triple negative breast cancer data set from BC Cancer Agency. There is a mutation, mutated acceptor site of the TP53 gene. There was coverage in the Intron. So the BC folks interpreted this as a case of Intron retention. We wanted to look at it. And so, yeah, look, there's coverage in this Intron, but the algorithm didn't say that there was an Intron retention. And we were wondering why. Well, it turned out that other introns downstream also had extremely high coverage. And the true story was that there's a mutation right here. There is a retention not just of this intron, but of all subsequent introns as well. So because our model allows for only one intron to be retained, we did not pick it up. Similarly, there are actually alternative acceptor sites, and there's also exon skipping caused by the mutation at the splice sites. We also looked at the case for a different gene where, and this was a data set from UNC, where they said they previously validated an intron retention. Our algorithm did predict it. But we also, when we looked, we saw some interesting alternative donor sites being used because there is donor site disruption, there were alternative donor sites, there was intron retention, and then there was also exon skipping. We gave all these genes and did a qT-PCR experiments. These were the abundances predicted by I reckon. This is what the qT-PCR reported. So there's some, I don't have their squared number here, but it's really nice. These are some of the other tools. So some of the isoforms were always missed by other tools. So finally, just to say, summarize, I reckon is a method for isoform discovery and gene abundance estimation. We can identify low abundance isoforms due to the use of proper regularization. So good computer science can lead to better biology. And because we've built it into a genome browser, you can visualize the splice variants. These are things which we could very easily go do and look at the data and not just sort of say, rely on the prediction from the algorithm. So finally, just like Leo, I'll skip some stuff and go to the most important slide, where I thank the I reckon team. So Aziz Mizlini is now a PhD student. He did this as his master's work. This was just recently published in Genome Research. The Savant team, led by Mark Fume, and this is where you can find the Savant browser. And this is the stuff I didn't get to. And finally, I want to thank all of you for listening. Right, so we do not explicitly look for differential expression. You could do it as a post-analysis step, where you go in and, given the expression of various, given two data sets, look at what's the expression of this isoform here and the expression of this isoform here, and compare them. But that's really not what you want to do, because it could be that here the regularization took us this way, here regularization took us that way, but it could have gone the other way in the two cases. So we do not do differential abundance estimation, although one nice thing about the Rna-Seq plugin is you can load two different data sets and look at their differences within the plugin visually, but not within I reckon.